Okay, let's start. Good morning, everybody. Welcome to our first webinar session of 2025, Tackling Listeria, a regulatory update and customer success stories. The guests' cameras and microphones are muted today to save bandwidth, but please feel free to use the chat for asking questions during the webinar. There will be a Q&A session at the end of the webinar, and our moderator will then pick and read the questions from the chat. And the presentation will also be recorded and made available for later viewing. So my name is Bob Meijer. I'm sales manager here at Tailor, and I will be your moderator for today. Today's agenda and speakers. First, we will have a welcome from Dr. Michel Schmidt, our CEO. Then we will go to the regulatory update, elimination of Listeria monocytogenes in the laboratory biosafety cabinet, elimination of Listeria monocytogenes at a ready-to-eat food production line. Then we will talk about free disinfection planning service. And then, as said before, we will have the Q&A session where we can have a discussion and some final words. I will now give the word to Dr. Michel Schmidt. Stage is yours.
Thank you, Bob, and a big welcome to everybody from my side. It's a pleasure to see that we are now, I think, 33 people joining. So this is very good because I think today's seminar is going to be one of the most interesting that we've had in a long time. I mean, we will discuss not only regulatory updates, which is of course important knowledge for all of us, but then we will have some really good real-life examples, how our devices have been used, and of course, very important in the end, we will show you a solution and how you can get this solution planning for free from us, because that really will show you how easy it is to implement these changes in your laboratories or production sites. But hey, I will not speak longer because there's so much more interesting for you to hear today. So without more words, I'm going to give over to our customer experience manager, Virpi Sorri. Please, the floor is yours.
Thank you, Michel. So my presentation today is about the regulatory update on Listeria monocytogenes and ready-to-eat foods. My name is Virpi Sorri. I work as customer experience at Tailor, and I joined this company in January this year. And before that, I have 11 years experience within the microbiology field as a technical support advisor in a bigger company. Here is the content for my presentation. We'll cover the current Listeria challenge, and then I'll explain background on the regulation and also the key changes. Then we'll discuss the possible impact on food business operators, go through some risk management strategies, and see what the expected advantages are. Then finally, we will discuss the recommended actions for 2025 before the change takes place. So let's start by asking, what is Listeria monocytogenes? It is an environmental pathogen that contaminates food. Listeria is found worldwide in soil, water, and animal digestive tracts. Listeria monocytogenes can cause a mild, non-invasive infection or a severe invasive illness, which is called listeriosis, and this has high fatality rate. This listeriosis is a high risk for vulnerable populations, including pregnant women, elderly people, and immunocompromised people. And also, what is special for Listeria is that it can tolerate various conditions, including acidic, dry, and salty conditions, with and without oxygen. Listeria also has an ability to form biofilms, which makes them tolerant to a wide range of different stresses, including UV radiation or chemical disinfectants. And this makes Listeria very hard to remove, for example, from production facilities. And Listeria monocytogenes can also survive and multiply in refrigerated foods. And this refrigerating survival makes ready-to-eat foods of particular concern. So what is the current Listeria challenge? Why a regulatory update was necessary? So the background is that there was increase in listeriosis cases. So in 2022, there was almost 16% increase in invasive listeriosis in EU. And this listeriosis has high fatality rate compared to many other foodborne illnesses. So for example, when compared to Salmonella doesn't cause that often hospitalization or deaths. So that makes Listeria a very challenging and also severe risk. There are challenges in controlling Listeria in ready-to-eat foods because they are a significant transmission route. There can be post-production contamination risks. So it is possible that Listeria enters the food from the environment within the food manufacturing facility. So therefore, there is a need for stricter monitoring throughout the supply chain. And previously, with the previous regulation, there was a gap. So the previous criteria were primarily focused on food producing operators, meaning the food manufacturers. But there was minimal regulation from factory to store, meaning that when the food leaves the food manufacturing facility, then what happens after that? There was no regulation present. Here is some data. So this is a trend of confirmed listeriosis cases in EU and UK from 2007 to 2023. And as you can see, the trend is really increasing. And of course, we know that the European population is also aging. So it is possible that these kind of keep on increasing because we have more vulnerable people in Europe. So that might be a reason why something has to be done through a regulatory change. So this slide presents the original requirements in the regulation from 2005. So the microbiological criteria is absence in 25 grams for ready-to-eat foods supporting Listeria monocytogenes growth before such products have left the immediate control of the food manufacturing facility. So this excludes foods intended for infants and special medical purposes. Alternatively, if the producer is able to demonstrate no growth throughout the shelf life, then the limitation is 100 colony-forming units per gram. The previous scope of this original regulation from 2005 was primarily applied to producing operators. So food producers were responsible for complying with this criteria. And there was limited coverage of supply chain stakeholders. So then what happens when the food leaves the food manufacturing facility? Therefore, there was also a gap in responsibility between production and final retail or consumption. And there was basically no monitoring throughout the product life cycle. So the food producers had the biggest responsibility to ensure that there is no Listeria present. But then what happens after that? There was no monitoring basically present. So here are the key regulatory changes. So the new regulation was published last year, 2024, and this new regulation amends the original regulation from 2005. The objective of this change was to enhance Listeria monocytogenes control throughout the food chain, and this is to strengthen public health protection. But through this new regulation, there is extended responsibility. So compliance is now required from all food business operators, including distributors, retailers, import or export operators who place ready-to-eat foods on the EU market. So the microbiological criteria are stricter. So if growth is possible, Listeria must not exceed 100 colony-forming units per gram throughout the shelf life. But if this cannot be controlled, it must be undetectable in 25 grams throughout the shelf life. So it is the food producer's responsibility to show that the limitation does not exceed. Otherwise, the stricter criteria is obeyed. And the new regulation was published last year, but it will be effective from July 1st, 2026. So there is approximately a year left before this new regulation takes place. So 1 year left to implement or plan actions for this change. So the technical criteria changes. So the core criteria remains. So it is still the absence in 25 grams for ready-to-eat foods that support Listeria monocytogenes growth, if producer cannot demonstrate the 100 colony-forming units per gram, that the bacteria does not grow above that. And that has to be scientifically demonstrated so that the national competent authority accepts this demonstration. The new requirement means that the criteria apply throughout shelf life, from production through distribution to storage and to consumers' hands. So this means that monitoring is required during the entire distribution chain, which also means that documentation of compliance has to be shown at each supply chain stage. Here are some of the food categories affected. So basically, the affected foods are ready-to-eat foods that support the growth of Listeria monocytogenes. High-risk categories are soft and semi-soft cheeses, smoked and cured fish products, cooked meat products, pre-prepared salads and vegetables, sandwiches and ready meals, also dairy-based desserts. And there are certain characteristics regarding pH or water activity, and also that the shelf life extended from 5 days onwards. Also, these foods are refrigerated and no further heat treatment is done before consumption. So here we discuss on the possible impact on food business operators. So for food producers, this potentially means robust shelf life validation studies and challenge testing protocols with live Listeria monocytogenes bacteria. Also growth, no growth validation studies, maybe temperature abuse studies, and all in all, better documentation. But now new requirements are also set for distributors and retailers. So they also need to monitor, which may mean like temperature control, shelf life management, and also documentation of compliance. There might be some operational changes needed, for example, regarding enhanced cold chain management, first in, first out protocols, but also staff training on Listeria risks, and maybe supplier verification procedures. Also, record keeping is important regarding temperature logs, product rotation records, supplier certifications, and also corrective action documentation. All this may significantly increase. And because there are the changed criteria and more food business operators impacted, this also means regulatory enforcement. So the authorities have more inspections and compliance checks to do. So here we discuss some risk management strategies, for example, through technical solutions. What is it that could be done? maybe reformulating products to prevent Listeria growth. For example, pH could be something, or packaging innovations. There are different type of packages that has modified atmosphere. Maybe new innovations in that field are needed, or processing optimization within the food manufacturing facility, for example, enhanced heat treatments. And then one strategy is regarding shelf life. Could it be reduced and that way make the food compliant with the new regulation? But then there are some operational solutions, for example, supplier partnerships. Maybe there could be shared responsibility through agreements. And then also cold chain excellence, continuous monitoring systems might be beneficial. And then, like mentioned before, staff competency development, so training programs for staff regarding Listeria risks. And all this also needs documentation systems. So as the documentation requirements increase, maybe there are some new solutions or digital tracking solutions that could be considered. So there are obviously expected advantages. So this regulation or the new regulation was introduced because something good has to be achieved through this. So obviously there are public health benefits, reduce Listeria monocytogenes contamination incidents within the food chain, which then hopefully also leads to reduced listeriosis cases. And then that brings enhanced consumer confidence when people can trust that the food is safe to eat. And then all in all, this creates stronger food safety culture. From industry point of view, the benefits could be equal compliance for all food operators. So not just the food manufacturers who are responsible, but this include all the other operators within food chain. This hopefully will also lead to enhanced supply chain transparency and improved risk management practices. And this new regulation could be an innovation driver for safer technologies because there is a need for proven Listeria control solutions. So hopefully there will be new technological solutions to improve Listeria control. Then as there is just 1 year left until this new regulation comes into force, here's what we think could be done now during 2025 and the beginning of next year. It is recommended to do an assessment, so conduct a gap analysis. What is it that has to be done in your food production facility? What are the gaps? What are the missing things? Also, identifying the affected product lines is part of this. And then it is recommended to review the current Listeria control programs and also identify who the key supply chain partners are and what is it that they are going to do. Then through assessment, it is recommended to do planning by developing an implementation roadmap, which also needs allocating necessary resources. And maybe the coming changes would also need to design new procedures and systems and also establishing supplier requirements. So where to get help at this point? We recommend checking the new EU regulation, which is available online. I have put the link here, but also the national competent authority has probably the local guidelines that they can share. I actually checked the situation with the Finnish Food Authority, and they have so far updated the guidelines on their website to reflect these changes. But at the time being, there were no further guidelines in pipeline, but they said that it might they might be doing then something when the deadline approaches. But then there are also industry associations, professional consulting services, and what is also maybe a good thing to do at this point is to network with industry experts. So you have colleagues in other companies. It might be a good idea to check what other people are doing in other companies and what is it that they're going to do. Okay, so this was my presentation about the regulation. Now I'm shortly showing you here our company technology as a bridge to the next presentation by Johanna Haavisto. So this is our technology. We manufacture spectral blue multi-wavelength high intensity technology, which is a chemical-free and UV-free method to eliminate listeria in production areas. As you can see from these pictures, we have real-life customers in different locations, different countries, who are currently utilizing spectral blue devices to eliminate microbes from their premises. So thank you for your attention. Here you can see my contact information. Feel free to email me or connect me in LinkedIn anytime.
Thank you, Virpi. And next up, we have Johanna Haavisto from Atria Laboratory. Johanna, please go ahead.
Thank you. So it seems it's my turn to continue. So my name is Johanna Haavisto, and I am a laboratory manager in Atria Finland. And I came here to talk to you about the elimination of Listeria monocytogenes in the laboratory biosafety cabinet. But I would like to start with a few words about Atria as a company. So Atria is one of the leading food companies in the Nordic countries. And we have been here for a long time. Our company was established in 1903, and we are valued by our customers, personnel, and owners. And as we have been producing the food for more than 120 years, and we would also like to continue for the next 120 years at least, we need to take care of our planet, people, and products. And as our main product is a good food, and it creates a better mood and sustainable value for our stakeholders. To give you some idea of our size, here you can see a few numbers. So our net sales in 2024 were 1.7 billion, and Atria had almost 4,000 employees in Finland, Sweden, Denmark, and Estonia. Originally, Atria is from Finland, and more than 2,000 people are working in Finland at the moment. And we are listed in Nasdaq Helsinki. You can change the slide, please. And on this map, you can see the locations we have. So I am located in Seinäjoki or Nurmo in Finland, but we do have other locations also in Finland and in Sweden and Estonia and Denmark. We are getting samples, or we are analyzing the samples from all these Finnish locations, and we have an accredited laboratory, and the scope of our accreditation includes all these microbial analyses that we are doing. But let's continue with the laboratory stuff more now. So why do we need the blue light in a biosafety cabinet? To demonstrate the safety of our products, we are doing a lot of pathogen analysis in our lab. So it's a daily basis work. We are analyzing Listeria, Salmonella, Stec, and Campylobacter. And we are analyzing these with PCRs, immunobiological methods, and also on agar blades. And alongside with the samples that we are analyzing, we are also cultivating the control strains, which we use to verify the suitability of the growth conditions of the bacteria that we are analyzing. And of these bacteria, especially Listeria, is capable of forming biofilms on surfaces, and thereby it poses a contamination risk in the laboratory. And these control strains are handled in the laminar flow cabinet or the biosafety cabinet. So it is crucial to eliminate Listeria from the cabinet. And for that, we are using the blue light. Please change the slide. But before we started using the light on a daily basis, we wanted to test its efficiency against Listeria. So we plated Listeria on metal plates, and these metal plates represent the surface of our biosafety cabinet. We didn't want to contaminate the surface of the biosafety cabinet as such, so these plates were much more handy solution that we could also analyze better. So we used our control strain, Listeria monocytogenes, and it was grown in brain-heart infusion broth, and we pipetted the 200 microliters of this dilution on the laminar flow cabinet. We wanted to use 200 microliters because we did a preliminary test where we were analyzing the growth of the Listeria, and we saw that with this amount we were able to get a good growth on the plates in the end results. So the control sample we were also having in another cabinet without the blue light to see how the Listeria would grow without the blue light. And we were using this blue light for 8 hours, which represents the time that we are using it in normal operation. So how we use this light is that we have a timer and we have it on through the night time. So on the left corner, you can see the illustration of how we put the plates on our biosafety cabinet. And you can also see how the blue light is located in the wall of the safety cabinet, and those plates are on the right side. And after the treatment, we analyzed the concentration of Listeria monocytogenes on these plates by plating. And you can change the slide. And we saw the growth only in the sample number 10, which was the control sample. So in all these samples from 1 to 9, there were no growth, and which was showing that this blue light was working well and after this test, we have been using the light and we have been happy with it. So that was our results. And I would like to thank you. And maybe you can continue with the next presentation.
Thank you, Johanna. It's really nice to see how you were able in the laboratory environment to show that the blue light did have such a phenomenal effect on killing the listeria. And it has been a pleasure working together with you on this project as well. So this was from the laboratory environment where we can show and we can test different things, how it works in theory, or not only in theory, also how it works in the laboratory as part of the hygiene control. But we also have customers that have tested and using it in food production premises. So the next case study is from a ready-to-eat food production line. My name is Karin Henricson. I'm here at led Tailor as the vice president and also in the sales team. So our next case, so this case is from a customer making ready-to-eat meals. And as everyone who has ever worked in the food industry knows, every now and then listeria can be found, especially on the floors. And so it was also in this case. So they found Listeria monocytogenes near a packaging line. It was primarily found on the floor beneath the packaging line. And of course, also as the operators walk alongside the packaging line, it's also likely that they can spread the contamination when they walk back and forth. They did try to eliminate the listeria with traditional methods, but they were not successful in this. So they decided that they want to test Spectral Blue. And they did increase the sampling before the installation and also after the installation. So there were 3 different stages in this test. In stage 1, that was before the blue light. So in July 2024, they selected 4 sampling sites on the floor and they increased the sampling frequency. In mid-October 2024, we installed 2 devices in one end of the packaging line, as you can see here in the in this picture here in the middle. Blue light was turned on by a timer, so always in the evening, and it went off in the morning. So they used approximately 14 hours per day. And the existing hygiene processes were left unchanged. We did see an effect already in stage 2, but to see if we could get rid, or if they could get rid of all the listeria, they decided for another stage, so stage 3, and that was in mid-January, they installed 2 more devices. So here we can see the sampling sites from above. So here is the packaging line, and here on the right side, we can see where the first 2 devices were installed in the beginning and during the stage 2. On the left, we did not have any devices, so that was a dark control area. The red squares are where they took the samples from the floor. And in the 3rd stage, we added 2 additional devices to the so-called dark area. And here we can see it also from a photo, what it looks like. So the devices are installed in the ceiling above the packaging line. And here we can see the results. So these results are an average from the different stages. So in the beginning, we could see that approximately 22% of the samples were positive for L monocytogenes. When we had installed 2 devices in one end of the line, it went down to a little bit above 10. And then we installed 2 more in stage 3, and it went down to 0 eventually. But in the, and for the first time, they could see weeks with 0 positive findings after installing the blue light. So this case study was accepted by and presented at the IAFP European Symposium in Madrid in May earlier this year. You can find the poster and also more data on our webpage, or you can be in contact with me if you are interested to hear more about this case. So thank you, and with that, I give over to Tuukka Autio, our product manager.
Thank you, Karin. So my name is Tuukka Autio. I work as the product manager at Tailor and Spectral Blue, and my talk will be about the free 3D disinfection planning service that we offer. So today's talks have been very interesting. We have got new information about the regulatory changes. We have heard 2 interesting customer success stories. And now my talk will try to tie this information up and show you how you can get this technical solution for the Listeria problem to use in your own facilities. So my talk is divided in 3 sections. In the first, I will tell you what does it mean, what do we mean when we talk about 3D disinfection planning. In the second step, I will tell you how it works process-wise, what do you need to do. And in the last one, I will show you how you can book it conveniently on our website to get started. So what is 3D disinfection planning? What does it mean? It basically means that we can do a computer-based 3D model of your whole food production facility, a single room, a set of rooms, or a single piece of equipment inside a room. When we have done the 3D model, we have digital profiles of all of our Spectral Blue disinfection devices, and we can just drag and drop them inside the model and see what kind of setup gives us the right kind of intensity and energy. So the computer software can run simulations with different setups and parameters. This, together with the vast amount of microbiological data that we have from academic studies on the efficacy of blue light and different tests with our own devices, combined with the years and years of experience that we have from working with blue lights and different microbes in different industries. Combining all these things, we can give you an optimal setup that is guaranteed to deliver results. That means when we do it this way, we will know for sure that there is enough intensity and the blue light dose is big enough that we can get rid of the target microbes. And when I say that we can do it focused on a certain part in the room, for example, production line, you can see that on the left. So this resembles the packaging line that Karin was talking about. We had 2 devices in one end of the packaging line. And on the right-hand side, you can see another option where we do a blanket installation and cover the whole room. And the decision is really based on what kind of microbe problem you might have and how you would like to solve it. So process-wise, how does this work and what will you get out of this? Well, it first starts so that you can go to our website, and we will send you this link via email, but it's easy to find on the web page as well. So you can fill in a simple form, give us the basic idea what you are looking for, and we will then be in contact with you to ask for more details if needed. And the basic information that we typically need is the dimensions of the room, how many hours can the blue light be on during the night, during the day, and if you know what kind of microbiological targets you are facing, is it Listeria, Salmonella, molds, biofilms, whatever. If you have blueprints or drawings, that's great, but it's not mandatory. Simple areas we can work with just the dimensions. And once we have got the data from you, our experts can make an initial plan for you. And this initial plan we can already spin on the computer screen. We can show it to you via Teams or during a visit. And then we can use your feedback to fine-tune the model. And at the end, as the result, you get a ready plan and a quote from us or from our regional distributor. And this includes pictures of all the rooms, list of Spectral Blue devices that you will need, the installation locations, and the recommended way of operating them. Is it with a motion sensor or a timer or a manual switch? And you will also get a breakdown of the cost per room, so you can decide where to start. And the planning service is free of charge, so there is absolutely no obligation to purchase. And also, if we notice that the scenario is beyond the capabilities of our blue light system and something else is needed, we will openly tell you so. So a little bit more about what we might need to make a plan for you. Well, the bare minimum set is that we need the dimensions of the room, length, width, and height, the blue light operation time during the day in hours, and if you prefer fixed installation or removable devices. If there's more data available, that's great. So are we targeting certain critical areas inside the room or the whole room? Is there a specific microbe problem like Listeria or biofilms? Or are you aiming for general disinfection? That's good to know. And if Spectral Blue will be the primary method for disinfection, or will Spectral Blue be installed to support the existing methods like chemical disinfectants? Here are a couple of pictures that show how the ready plan will look like. On the left-hand side, you can actually see the RTE production line case that we simulated with the computer first. So the computer gives us this heat map of the blue light intensity, so we can know for sure that there is enough intensity to do the work as intended. On the right-hand side, you can see a complete clean room facility that has been planned in the same way. So this is my last slide, how to get started, how to book the free planning. So please proceed to this address. There's a small form where you can give basic information about your company, about your facility. And if you want to leave some details, there's a comment field for giving us more information. And like I said, we will then contact you and ask for any other details that we might need to make the initial plan. So thank you very much. That concludes my presentation, and I will hand it back over to the moderator for the questions and answers.
Yes, thank you, Tuukka. Let's see in the chat, there's a question for Virpi. So the regulations mention controlling the Listeria growth. What mechanisms are available or in use besides refrigeration?
Yeah, well, there are two sides. So it is a good idea to control the Listeria in the raw material within the food itself, but also in the environment, that it doesn't come from the environment. So, you know, for example, you can play with the formulation, for example, regarding pH, like I said. said, so Listeria growth is generally prevented at a pH lower than 4.4. And then there is also water activity involved. So, drying the food might also be a solution, but this would depend on what the food is. What is it that you're manufacturing? But then also there are like process related things what you can do with the food manufacturing process, like heat treatments and so on, or the food packaging. But then also environmental control, so enhancing hygiene within the environment so that the Listeria from the environment doesn't enter the food. So I think I would start with that. And our Spectral Blue solution works for that purpose.
Okay, thank you. Another question in the chat. Does the blue light only have an effect on exposed surfaces? What about hidden surfaces?
I can start about that. So that's a common question that's typically many people ask, but what about shades? What happens in the shadow? Well, everywhere that you can see with your bare eyes, there is some light, at least. If it's completely pitch black, then there's zero light entering. Typically, blue light will reach areas where a human hand, the hand of the cleaning person or manual disinfection person doesn't reach. So in any case, the blue light will go into areas that are otherwise not accessible. But it is true that the less intensity we have, the longer time we will need to disinfect those areas. So I would say that we have to take that into account in the planning, to be aware of difficult areas where there might be large shadows. And we have to see that if a device can be brought closer, or if we can use some other way to bring enough blue light into these areas.
Okay, thank you, Tuukka. Another question, how are biofilms affected by the blue light devices? So what if the Listeria is in a biofilm?
Yeah, let me start with that as well. Thank you. As we know, biofilms are really difficult to eradicate with traditional methods because chemical disinfectants in normal concentrations do not really penetrate the protective layer of biofilms. And the same goes with UV radiation, that it's easily absorbed into all kinds of materials and doesn't really penetrate the protective layer. With blue lights, luckily, the blue light is capable of penetrating a lot of materials like water, glass, plastics, and also the biofilm layer. So the blue light can actually go inside the biofilm and affect the colonies inside. For sure, based on our own tests and literature, it will require a higher dose. So you will need a longer time than with regular colonies that are not protected by biofilm. But if you use blue lights in a continuous fashion, you have it operating with a timer. So over time, you will accumulate enough dose to eradicate the biofilms. And after you have eliminated the biofilms, it's really difficult for the microbes to regrow the biofilm because there's always blue light suppressing the growth and preventing them from generating these biofilms again.
Okay, thank you. I don't see any more questions in the chat. If anyone still wants to ask a question, now's the chance. What about hand disinfection? Can you maybe elaborate on that question a little bit? Do you mean, for example, how spectral blue disinfection compares to manual disinfection in terms of efficacy, or what? Can you maybe put in the chat what you mean by that? Yes, Kirsi.
So good afternoon, everybody. So if I understood correctly, this question concerns about the hand disinfection and blue light, if this could be used as a non-chemical hand disinfectant. And if this was the question, so I have had been in a couple of studies where we studied if blue light could have been used in hand disinfection, but it's not very easy since the hand disinfection should be performed in a couple of, well, let's say less than 1 minute, preferably 30 seconds. And at that time, it's not possible for blue light to do the disinfectant. And then again, it needs some kind of supporting agents like some chemical agents to be added to the procedure, and this enhances the efficacy of blue light in skin disinfection, but so far, it does not. The blue light is not as efficient as, for example, the alcohol disinfection.
Okay, thank you, Kirsi. And thank you for asking the questions in the chat. Were all the answers satisfactory? Because if there's no more questions in the chat, I think it's time to wrap up. So thank you everyone for attending, and please let's stay in touch. So just to sum up, you can request a free 3D disinfection planning at our website, spectral.blue/free-planning, or contact us at info@labtailor.com for more information. You can follow us on LinkedIn for all the latest news and events. It's linkedin.com/company/spectral-blue. And please check out the video recordings of our autumn 2024 webinars as well on our website, spectral.blue/webinars. So there's a series of 5 webinars that we hosted in the fall, and we posted on the website. So the first one is an introduction to Spectral Blue multi-wavelength high-intensity blue light, if you want to know more about the science and application behind our devices. The second in the series is the application for clean rooms. There's the application of replacing UVC and laboratories. The fourth one is application for the food industry. So there's a bit more information relevant for this public perhaps. And the fifth one in the series was healthcare and ambulances. And you can always send one of us an email or reach out on LinkedIn as well if you would like to know more. So thank you all again for attending, and let's stay in touch.